more experienced friends than me because it's been 1 year and I haven't yet visited the bakery you're talking about. So I need to go there once the lockdown is lifted.
It's very close to the university, so.
Okay, well, okay, I need to get out of the lab. Okay, well, with that, welcome everyone and to the IMPMC Mineral Life Seminar. My name is Neha. I'm a postdoc with Kareem, and it gives me pleasure to organize these seminars along with Elodie Mueller, who is also a postdoc in Biomin team at IMPMC, and Elodie Dupret, who is an associate prof at Sorbonne University. Both of them are in the participant list. First of all, I want to thank all of the participants because I understand it's a tricky times like right now, and I really appreciate you all taking time to join us for the seminar. With that, I would like to welcome Jeff, especially for waking up at 7 a.m. to give us this talk. Jeff is assistant prof at University of Colorado, Boulder. He is also a fellow at Renewable and Sustainable Energy Institute and has a joint appointment at National Renewable Energy Lab. His group's research, from what I gathered, at least from brief description, focuses on using cutting-edge synthetic biology tools to investigate the metabolism of the single cell, as well as biochemistry of photosynthetic cyanobacteria. That sounds really cool. But today he will be talking about a topic which is very close to the ongoing research in quite a number of groups here at IMPMC, which is role of cyanobacterial CCM mechanism on biogenic carbonate precipitation. And with that, Jeff, I'm going to hand it to you. The floor is yours.
All right. Well, thank you. So, pleasure to be hosted here, and thanks for the introduction. So I would say, so my lab is very interested in kind of the architecture of microbial metabolism, and in particular in the carbon concentrating mechanism and carbon fixation. And it turns out that kind of the ancient lifestyle of the cyanobacteria and all the things that they're involved in with in Earth's geochemistry has really kind of got me involved in lots of different projects. And one of them is in these calcium carbonate precipitations. And, in fact, when Kareem came here several years ago, that was, I've been kind of interested in it, but I think he really showed me the beauty of what these things look like inside of this cell. And I've, now spent quite a bit of time looking at these extracellular carbonates. So let me see if I can jump into this slide. So I'm going to get to the carbonates at the end of the talk, but I want to kind of give you an overview of kind of the thought process in my lab and what I really have set out to try to do in terms of, kind of coming out as a plant biologist and then studying cyanobacteria for the past 15 years. And when I, finally got my own lab space, I really wanted to take a different view and really the single cell view of cyanobacteria. So I'm going to give you kind of a big picture of my big interest, and then really a little bit about what I've spent the last 5 years doing is really the hardware software tools for really like high throughput understanding of biology. And then I'll tell you about that biology in a second. So the kind of the key thought of my lab is really thinking about the structure function relationship of biochemical processes and metabolism, but really in the context of its location, and that location being in space and time, because that's critical to provide the correct environment, the right protein-protein interactions. And so really these, the structure function location is this, I think, critical concept to understand, how a photosynthetic cell can do all the processes required for life in this kind of 1 micron by 1 micron box. So this little box that I'm very interested in is this, let me note, sometimes my slides are big and the movies are delayed. So if ever you feel like my presentation's messed up and I just keep on talking, feel free to jump in and say stuff's messed up. So, okay, so this is the kind of one of the key model organisms in the lab. It's a unicellular photosynthetic cyanobacterium, Synechococcus 7002, a marine cyanobacterium. And this is really the model that we use to study growth because it has a lot of history about it, a lot of knowledge, sequenced genome, cultivation, lots of tools. And I think it's one of the most simple types of organisms, really, just to study the process of growth. And so this is we can grow it on light and carbon dioxide as its sole energy and carbon source, and liquid and plates. And importantly for my lab, we're interested in kind of the context of everything in the cell. So this is a electron micrograph of a dividing cell where you can see the internal thylakoid membranes. The cell's just about to divide by binary fission. So that's where all the photosynthetic reaction occurs on these internal membrane systems. Here's a large inorganic polyphosphate granule. So this is important for a lot of processes. And then these are these carboxysomes, which are these protein-based organelles. So all of the carbon fixation in the cell occurs in these structures. And we do a lot of kind of visual representation of cells, so by electron microscopy or various live cell imaging. And so this is kind of looking at the same view of the cell by super resolution imaging. But it turns out the photosynthetic complexes in the membrane are fluorescent. So here you can see the red are the fluorescent pigments, chlorophyll. We have the nucleoids stained in blue, and the rubisco, the key carbon fixing enzyme, fused to a GFP, where here we're showing it by immunogold localization. Here we're showing it by fluorescence, where you can see all of the carbon fixing capacity of the cells is spatially localized within these individual foci. And this has a lot of implications for kind of the overall fitness of a culture and heterogeneity of metabolism. And importantly, these organisms take light energy. They have this specialized light harvesting complex termed the phycobilisome, which transfers energy to the reaction centers, allowing them to split water, produce molecular oxygen, and then eventually fix carbon dioxide into the sugars. And as I'll tell you about, this process of the carbon fixation occurs within these specialized structures. And overall, cyanobacteria, as many of you know, are important global primary producers, in the oceans, lakes, terrestrial environments, and the carbon fixed by them accounts for about 30% of the carbon fixed each year. So the cyanobacterial physiology is very important and globally relevant. And over the past few billion years, they've also greatly influenced global geochemical cycles, as we'll talk about. Okay, so just kind of a brief overview. I'm not going to talk about all of these things, but in general, the lab is really interested in kind of, where are all the processes occurring and why are they where they are, and is this important for the function? So we've spent a lot of time looking at gene expression, RNA turnover, and now we're starting to learn about how RNA in the cell can actually direct processes to spatial locations in the cell. We do a lot of work on kind of the organization and the dynamics of the photosynthetic membranes. And then I'll spend more time talking about these basically specialized protein-based organelles where enzymes are encapsulated in a protein microcompartment in cyanobacteria. And these properties have really given cyanobacteria some of the most kind of amazing metabolic capabilities. And in fact, they're the only organisms that are able to perform oxygenic photosynthesis, carbon fixation, and nitrogen fixation, really through the spatial and temporal separation of metabolism. Okay, so this carboxysome, which I'll spend a fair amount of time talking about, is this protein-based organelle that resembles an icosahedral virus particle. And so if we look at the chlorophyll to outline the cells on the left, and we fuse the rubisco, this carbon fixing enzyme I'll tell you about more. If we were to look at that, this is really defined by a shell architecture. And it turns out that this special shell, it's a protein-based shell that allows passage of metabolites into the interior. These shells are related to a whole class of protein-based organelles found in bacteria. Kind of the most well-studied would be the carboxysome. I'll tell you a little bit about these shell proteins, but really the key is that these are really allow you to control the flux of gases across the interior through this specialized pore in these kind of self-assembling icosahedral particles. Okay, so, kind of just going back to this, in this case, this cell on the left is just about to divide. It has 4 of these particles. Each one of these has about 2,000 Rubisco molecules. If this cell were to divide, the cell on top would have, a quarter of the total Rubisco and would grow slower than this other daughter cell. So this essential carbon fixing enzyme is not evenly partitioned between daughter cells. There are mechanisms by which cells try to do this, but in this case, the heterogeneity of just due to partitioning of these could really have influences on growth. So we're really kind of interested in how that's manifested in a population. And so the key concept that we're really looking at is this single cell resolution is essential because the ensemble can mask a lot of important features. So let's just say you're trying to produce a purple pigment or any sort of trait, and you look at kind of your theoretical max and min. If you have every cell producing at 100% versus your blank control, you could use an ensemble measurement to distinguish between these cultures. If they were perfectly homogeneous, you wouldn't actually have to measure the individuals because the average kind of represents the population. But the reality really is in almost any microbial population, you can get cases where half the cells might be producing at 100%, the other at 0, versus the entire population might be producing at 50%, the max, for example. But the average of these populations would actually be identical. But it turns out if you were wanting to get to this population on the left, your strategy might be very different based on what these averages are. And so we think it's really critical to be able to look at every cell in the population to see what it's doing, and we can actually use this as a tool. And it turns out in cyanobacteria, this can be almost inherently generating this heterogeneity because light is effectively a 2-dimensional nutrient. And so if you have a thick culture, over time, as the culture gets denser, they will shade themselves as a function of their own density. And so in a batch culture, you really can never achieve steady state because of this continuous attenuation of light as they grow. And this can generate heterogeneity in a bulk culture. And if you're really interested in kind of, keeping track of energy and carbon, not knowing how much substrate the cell gets in terms of energy can really limit your understanding of, its ability to use that energy for performing work. So to get over that, we really wanted to shrink this volume down to the size of a single cell thickness, and we can overcome this shading and heterogeneity. So with the next few minutes, I'm going to tell you about kind of the hardware that I really wanted to develop when I started my lab to get this to work. And I would say kind of the first thing, and I won't talk too much about this, is having really robust, high resolution, electron microscopy, super resolution images. This really gives us the best view of the cell. If you look at this left, this is a electron tomogram showing virtual nanometer sections through a cell. So this is really how we like to view the cell, and it can teach us a lot of things. So actually, we can see these polyphosphate bodies, are these large granules here. The carboxysomes that I'm telling you about, which often are represented as perfect icosahedral structures. Here you can see a very large one here, overlapping with a few other ones. I can play this movie again, and you can see that they're often sitting at these specialized corners of the membrane. There's a lot of organization of where these structures are. So this is kind of the visual, but this is takes a long time to produce one of these images, and it's a kind of a static picture here. And so we wanted to be able to do a lot of this in live cell imaging. And this required kind of a little development of technology, because most of the kind of tissue culture facilities used for mammalian imaging are not really sufficient for doing live cell cyanobacterial imaging, because they use CO2 and light as their energy and carbon sources. And if you're imaging with light, you're having to kind of keep track that you're using light to image them, but it's also their growth light, and this can be very toxic, for example. So the key is that when I started my lab, I built this specialized microscope where we grow our cells either in a microfluidic device or on in between an agar pad that contains nutrients and a cover slip, restricting cells to a two-dimensional layer. We can then provide light, which is then spatially and spectrally controlled from above, so not just a white light, but we can actually control our RGB and make white light. And it turns out cyanobacteria can sense the red to blue to green ratio and also the intensity, and this is very important for the cell's physiology to be able to control the light. The environmental chamber, we can control the atmosphere, so the carbon dioxide concentration, oxygen, nitrogen concentration. And then we're imaging the cells from below, and we can use different either excitation or emission filters then to capture information on the photosynthetic pigments or other activities of the cell. Importantly, we're using light to grow the cells as their sole energy source, and then we're filming them, and kind of getting the hardware and synchronizing this process was really key to be able to get long-term steady-state growth conditions going over, now upwards of over 11 days where we can film cells. And the cool thing about this system is that if we have adequate CO2 and nutrients, light can be our only limiting nutrient, such that we can titrate growth rate only by manipulating the photon flux that we're providing to the cell. And this allows us, a lot of tunability of growth rate independent of any other condition, basically allowing a kind of a light limited chemostat condition, which is really only possible in a two-dimensional layer. Okay, so can I get a heads up? You can see the start of this video. This is where I often, so here what you should be seeing are 3 cells sitting on a black plane. Is this what we're seeing here, hopefully?
Yeah, that's right.
Okay, so this is now just taking a view of zooming in on 3 cells that we've taken from a asynchronous liquid culture, spotted them onto an agar pad, and now we're filming them, and we're synchronizing them by turning on the light. And I think this is just to show really that once we actually do all this growth in the microscope, it's extremely reproducible, not only in terms of the growth rate, but also even the colony morphologies. So here, we're filming the photosynthetic membranes as the channel that we're looking at, so chlorophyll here. And here, these are 3 microcolonies that have 16 cells, so this was after 4 doublings here. And these are 3 independent microcolonies from different cells. And if you were to zoom out, almost every colony actually even grows to this identical colony morphology. I'll just play this one more time, because many people think that this is kind of a copy of 3 different movies. But it actually just turns out that really this is driven by, almost purely mechanical properties here. When you turn the light on, the cells expand, they fold themselves in this particular position. We know a lot about it, and I'll tell you a little bit about it now. So once we can capture these really good films, so that was over about 16 hours, and we can get lots and lots of other cells in that one image. The goal is to be able to film those cells and then extract information through cell segmentation. This is getting the computer to identify a cell. We then can do data measurements on those cells, track objects over time. The goal is to be able to store this data in a really kind of a multi-dimensional database that we can use for data mining, machine learning, things like that. But really the key is to extract high quality denoised data and then image it. I won't tell you too much about this on the right, but it turns out the key is really, you can see a cell easily. You saw those cells before. I didn't have to explain what a cell was. Getting the computer to recognize a cell is more difficult, and we're doing a lot of kind of machine learning to do this. I'll show you some of the results of this. Let's see, I might have, sorry. I skipped ahead. I might have missed one slide. Okay, so we have I don't have the citation below, but we have a toolbox called CYAN, which is basically a MATLAB-based toolbox for automated imaging. So all of this is now published. There's a link in a few slides that actually has the link to the bioRxiv paper on this. But the key is that the original kind of algorithms, generating these masks and over these movies still takes a lot of manual curation to really, there's a labor of love to look through 2,000 frames of a movie, make sure there's not a pixel connecting cells, which can give you messed up readings. But once we did that for about 20,000 cells, we were able to use that to train a bunch of deep neural networks So now we have a new program. This will be now added to the bioarchive thing, but this will actually be very useful. So this is a deep learning neural network to automatically detect cells, even in very complex bright field images on the left. We validated this as it's greater than 99% accuracy on ground truth curated human data sets. This is on a never before segmented image, where here is a bright field on the left, and then on the right is basically a yellow. This is a binary mask where yellow equals cell, purple equals background, basically a prediction of cell. And it turns out this is good enough where you can just apply this and do this in about 2 minutes, and it overcomes the need to do any sort of manual curation just based on how much data you can generate. So this, it will allow kind of real-time analysis in the movies. But at this point now, what we want to do is we can film the cells on the left, in bright field, for example. And then we can, computationally extract out all of the cell masks. Hopefully this movie played here. And at the end, you should see, that now each one of these cells has a number in it, and we can now keep track of every one of those numbers as a unique cell ID to ask questions about the length over time of cell so-and-so, or the history of each one of these cells. So this is a way to basically store all of the data for every cell in the movie. And once we have those cell masks, then we can actually easily, ask these questions, for example, of, cell length over time of that mask. We can fit this to an exponential curve, and then, find the growth rate of an individual cell, from the movie. We could look at other traits like chlorophyll fluorescence, for example, or count dots. In this case, this is looking at chromosomes, so cyanobacterial polyploid. Each one of these dots represents a chromosome. We can extract out and count dots over time, for example. We can also count carboxysomes, for example. It's really a foci counting algorithm. And so this now gives us these tools to kind of count single cell phenotypes. And then the thing that I've been most excited about lately is, a way to now represent this complex information in what's kind of termed in computer science as a binary tree. So we can map all of those information, the growth information, onto these kind of tree plots here. So on the top right, it's showing a single cell 1 dividing into daughter cells 2 and 3. Cell 2 divides into 4 and 5, and 3 into 6 and 7. We can basically now keep track of that on this tree, where this branch equals the length of time that cell 1 was alive before dividing. So this branch length gives you basically the time to doubling. And then at each one of these diversion points, this would be 2 different cells. And once we actually get the data in the structure, there's a lot of algorithms to just traverse this tree and extract information, such that we can easily just pull out from this tree the growth rate of cell 1, 2, 3, 4, 5, 6, 7, over this 12-hour movie, and compare between lineages, colonies, or cells kind of in any position. And one thing that's been really informative and unique is it turns out that this colony morphology is so reproducible that it actually preserves the lineage information within the colony architecture itself. So in this case, we can, like I mentioned, we can computationally pull out growth curves across a tree, but it turns out even if we don't film the cells, because they always go into the same position, we know that cell number 16 came from 8, 4, 2, and 1, and it preserves, the old and the new pole based on their bending, which has a lot of interesting properties. And it turns out, like, using this, we actually found that the cells in the interior are actually mechanically confined, because in a 2D layer, they have to push all their neighbors out of the way to physically expand using light energy. And this leads to an attenuation of growth in interior colonies. You can start seeing it here, where cell 9 will actually start growing slower than its outside neighbors. So if you were to just average the growth of this colony, you'd be biasing it towards position. But using like the symmetry in this colony, you could compare 16 and 31, 17, 30, 18 and 28. These are these symmetric pairs, which really allow you to analyze growth in a very sophisticated way. So I spent about 2 years on kind of this high risk, high reward project to figure out now, can we film the cells? And I really wanted to capture what do these cells look like by EM. And so we basically figured out how to film the cells, plunge freeze the slides, embed them in resin, section through them to find the layer of the cells, and then turn them and face them up to get an unfaced section of through a single cell, 16 cell microcolony, where we actually know the relationship between every cell in this colony. So I'll kind of flip back and forth a little bit, but if you were to look at kind of the typical colony, these are grown on actually stiffer media, which actually can attenuate the cells in the interior, which you can actually see the cells in the interior, they should be synchronously dividing under optimal conditions. Here they're square, and you only get growth on the edge colonies, so cell 20 and 23. And so if you look back here, you can actually see that the cells in the interior are square. There's a lot of kind of interesting thing on the membranes. So these are the thylakoid membranes. There's actually a symmetry. That is due to actually kind of an epigenetic inheritance of membranes that we found here. That where actually these cells diverge after the first generation, but this asymmetric membrane structure is actually preserved for 4 generations here. And you can see a lot of kind of other interesting structures here. But I would say kind of the end kind of thing concluding this section is it turned out that understanding the mechanical forces between cells was a critical thing to actually understand how to grow cells in the microscope. Much of the artifacts and cell death that we experienced over 2 years was due to mechanical confinement leading to high light sensitivity. We've published this in Nature Microbiology recently, but this is now showing a simulation of a mathematical model that we've made to basically simulate those growth properties of these cells. And it's really, this is a ODE model of growth with also the incorporating the Newtonian mechanics of cell-cell interactions, the elastic modulus and kind of elasticity of cells, and then kind of the frictional interactions between cells and the media. And really it's this kind of pure mechanical model can reproduce this. And it turns out actually that these cells in the interior, which we know are eventually mechanically confined and get squared off, shown here, eventually we can, if we measure their photosynthetic activity, we've found that they repress their photosynthesis. So here they start off in this low fluorescence state, high photosynthetic state. But as they get confined in the interior, this kind of brightness is them dissipating energy when they can't expand in the middle of a colony, because it turns out they're still harvesting light. They cannot move. They have photosystems, but they can't physically expand because of their neighbors. And this will lead to a dissipation of this energy and a change in their metabolism. I'm going to switch directions a little bit here, but I'm going to come back to this because I think when people are thinking about microbes in their environment, and especially in mineral environments, mechanics is key. And it turns out this mechanical feedback, really probably drives a lot of this changes in metabolism, which can actually lead to mineralization, sometimes pathogenic mineralization, and these other biofilms like that are pathogenic. So I think this kind of concept of repressed growth mineralization is something to think about. Okay, so with the last 15, 20 minutes of the talk, I'm going to kind of switch now into really kind of my passion in carbon fixation. I'm going to start in deep time because I think it's really important to think about cyanobacteria in the context of Earth's history, and then what they're doing today, and really what we can kind of think about, how they're contributing to production of minerals and carbon sequestration over geological timescales. So cyanobacteria, have been around for somewhere around over 2.5 billion years ago. They're attributed to generating oxygenic photosynthesis somewhere before what's termed the Great Oxidation Event. And this is very substantial. So it turned out the ancient Archean Earth was high in CO2, low in oxygen. So it was this anaerobic Earth. Cyanobacteria came along and basically evolved oxygenic photosynthesis, allowing them to produce molecular oxygen, leading to the rise in global oxygen levels. Over time, CO2 levels have dropped, but much more slowly than really the rise in oxygen. And I won't have time to go into this other kind of whole other story, but we recently have really done a lot on kind of looking at the preserved isotopes during this time period. We'll have a paper that should be coming out very soon, but it really kind of suggests that cyanobacteria with a carbon concentrating mechanism actually dominated productivity during this kind of steady state period. And what I'm going to talk a little bit about and jump ahead a little bit, but the cyanobacteria contain a carbon concentrating mechanism. It's termed the carbon concentrating mechanism. But what we actually found is that it's much more likely this evolved in response to the great oxidation event to actually exclude molecular oxygen from interacting with rubisco. So here's the kind of key thing to think about this context. Cyanobacteria were fixing carbon and thriving in this low oxygen environment, high CO2 environment, and then the atmosphere changed. And actually this represents about 100 million-fold increase in the oxygen to CO2 ratio, even at 100-fold levels of CO2 to the current. And it turns out rubisco, the key carbon fixing enzyme, which was present in cyanobacterial ancestors, all of a sudden faced this kind of unusual selection pressure where the cells need to fix CO2 to grow, but they also need to produce oxygen for photosynthesis in the same cell, setting up this competitive situation where it turns out rubisco can use oxygen or CO2. It's a carboxylase oxygenase. The preferential reaction is to fix carbon dioxide, and it turns out if you fix oxygen, you generate a toxic byproduct and you have to recycle this CO2. And so once oxygen was produced in the atmosphere, this set up a competitive substrate for rubisco, and rubisco is then evolved to either optimize growth of these different organisms. And it turns out there's a trade-off between either the maximum carboxylation rate of rubisco or the ability for rubisco to discriminate between these substrates, so the specificity factor. So either you can be very specific, such as the red algae, and preferentially use CO2 over oxygen, but at the expense of carboxylation rate, so the speed of carbon fixation, or as plants have done, you're at the sweet spot between specificity and carboxylation rate, and they have a lot of systems to detoxify this oxygen byproduct. Or in the case of cyanobacteria, they've maintained the fastest rubisco at the expense of this specificity factor. So cyanobacterial rubisco is a very good oxygenase, very sensitive to oxygen. And it turns out they can do this because they've co-evolved the CO2 concentrating mechanism, whereby they accumulate bicarbonate from the environment. That bicarbonate then is trapped, it's charged, so it can't cross the membrane. It could enter the carboxysome, where it's converted to CO2 by carbonic anhydrase, which is only inside the carboxysome shell. And then rubisco then, which is now saturated by rubisco, can fix basically CO2. And what we're starting to learn is that this shell is really excluding oxygen from this region, allowing rubisco to basically be a fast carboxylase and not have to worry about its own specificity factor, because that is taken care of by this external shell, which controls oxygen permeability. And importantly, the shell is actually conserved across all known bacterial microcompartments, a whole class of them, with the carboxysome being the only anabolic reaction chamber building up things, so fixing carbon dioxide. There's a huge class of anabolic chambers breaking down substrates like alcohols, propanediol. Here's an example from Acetobacter. Really the key is that the shells are conserved, but the enzymes encapsulated are not. There's very different. But the key feature is all of them contain oxygen-sensitive enzymes. And so really this is pointing to this as an evolutionary adaptation to the rise in oxygen to allow you to do effectively anaerobic metabolism inside of an aerobic cell. Okay, I won't go very detailed into this, but it turns out, so we've been able to make a mathematical model of this system, really kind of a simplified system to look at gas flux and equilibrium between the atmosphere to the water across the cell, which requires a lot of different carbon transport systems, and then into the shell. And it turns out that really the only thing that matters is that rubisco has a lot of CO2 around it, so the cell can grow. This requires this active carbonic anhydrase. If we knock out this carboxysome shell, we can have rubisco, so it's freely soluble in the cell, the cells will die in air, but we can keep them alive at high CO2. So just by elevating the environment, allowing us to really understand both these systems by just controlling the atmosphere, we can generate mutants. When you look at wild type cells, if you were to label carboxysomes, you'll see that here's hundreds of Synechococcus 7942 cells. So these are a common organism people study. Each cell has about 4 or 5 of them. Each one of these dots represents 2,000 Rubisco subunits in this structure. But here you can't really tell, is this an active one, inactive one? Even with an electron microscope, you couldn't tell what came before and after, or with light, you can't tell, based on the resolution of these 200 nanometer structures. So really looking at this in a population, it's a needle in the haystack, which prevents a lot of mechanistic understanding of what's going on. So just like I mentioned earlier that we want to look at single cells, we also want to look at single organelles within the cells, because each individual one of those carboxysomes could have their own activity. One could be old, one could be young. And can we resolve this? So we developed a tool then to now control carboxysome expression and a computational framework to now look at single carboxysomes in single cells. I'm gonna jump through some of this because I wanted to show you guys some of the crystal pictures and some of the implications, and we can always jump back if there's questions. The key is that we can delete the native system, keep these cells alive in high elevated CO2. We can put the system back with an inducible operon using synthetic biology and make these strains, whereas wild type or with Rubisco labeled, they can survive in air or high CO2 with and without inducer. If we knock out the CCM, these cells cannot survive in air at all. We can keep them alive at high CO2. But this inducible strain nicely dies in air, but we can perfectly rescue it in air with inducer, showing that we can go from this state to this state just by turning with a chemical. And this shows that we can also image this process. So if we were to look at kind of the key one on the bottom here, we can go from a state of soluble Rubisco to encapsulated Rubisco. This correlates with growth and functional expression of carboxysomes. So here's just a video now where we can do a time lapse of imaging the de novo formation of carboxysomes. So you can see a cell can't grow in air until it makes its first one. Now we make a lot of them, and the cell, enters a steady state growth, and we can grow. Hopefully, can we see a movie? Hopefully. Okay, good. So these are cells with a lot of carboxysomes. It's kind of madness. But what we find is that in that case, there's really too many to count, so what we did is figure out how to induce them and then wash out the inducer, so they can only make a few and then can't make any new ones. And now by tracking them, we can find that cell number 3 can't make any new ones. It only can fix carbon with that one organelle. It can divide to produce 2 cells, but cell 5 can't fix any more carbon in air because it doesn't have a carboxysome. Cell 4 can, and we can track this now over time and basically look at these lineages. So this is now an example of using dilution in a population to measure the activity of individual carboxysomes by relating it to the growth rate of its individual cell. So in this movie, let's play it one more time. Here you can see now this cell only has a single carboxysome. Only the cell that inherits it is able to grow. The other cells can't expand. And at the end of its life, we actually can see it gets recruited to this place and turned over. So this was very valuable because in bulk, we can now dilute carboxysomes over time to less than 1 per cell in the population. And we can count the number of dots in every cell. So we can measure now the growth rate as a function of carboxysome number in the population. And importantly, in this cells with only 1 carboxysome, the growth rate is proportional just to that carboxysome activity, because that's the only way they can fix carbon, accumulate biomass, and grow. So we developed kind of a mathematical framework to extract all this info from movies. Basically, we can just add the sum of lengths of a cell. So this is, very simply, how much total biomass did a cell produce with a single carboxysome? Because they're only expanding basically as in this 2 dimensions, right? They're expanding and dividing. They don't expand in width. So we can basically measure the length over time and get a lot of information. So in this case, if we take all of the data from these movies, or we can get hundreds or thousands of individual carboxysome traces, we found that they cluster into these 3 distinct traces, either ones that never grew, ones that the cells were growing and immediately stopped growing, which was followed by a degradation event of the carboxysome, or cells that can continue to grow. Kind of very briefly, it turns out this no growth phase is correlated to a change in position in the carboxysome to the pole in the cell. So when they become inactivated, they are recruited to the pole and then degraded shortly afterwards. I won't have time to tell you about that, but now we know quite a bit more about this degradation pathway. It turns out even a cell with 2 carboxysomes, here the cell, 1 carboxysome lost activity, we can see an inflection in that growth rate followed by a loss of 1. So now this cell is growing at the level of a single carboxysome from 2. So the cell can sense an inactive carboxysome even in the presence of also functional carboxysomes. And if we look at some of these other kind of growth traces, I'll kind of show you what they look like. So in this case, the cell had 2 carboxysomes. It got diluted to 1. Here the cell is dividing, but its daughter cell couldn't divide. And so these are only the carboxysome lineages where the cell is growing slower over time. So this is following an exponential decay of activity where we could get the half-life of a carboxysome activity over the population, which is about 12 hours, which is much longer than the doubling time of the cell, which is about 3 hours, suggesting that these are contributing to growth over multiple generations. Kind of interestingly, we found another class of these ultra-productive carboxysomes where this is a case where a cell lineage with a single carboxysome is keeping constant growth rate for 7 generations with a single carboxysome. So if we follow this is now a cell with a single carboxysome generating, daughter cells, in this case over 7 generations. Now we've actually seen a single carboxysome over 17 generations, over 11 days, still remaining active, and we can generate, in that case, a lot of daughter cells, about 85 daughter cells or so. This slide is a little pixelated, unfortunately. I think I put the wrong image on here. The final thing I'm going to show before I jump to the crystals is that we really wanted to kind of prove that it's the carboxysome activity itself that's responsible for this growth. So what we did is we grew cells and diluted the carboxysomes under ambient CO2 where they're required to generate these conditions where, if you didn't inherit one, are those cells dead or are they just sitting there? What's happening to them? And then we were able to add elevated CO2 after 42 hours of generating all these classes. On the bottom, it basically shows that every one of these cell classes we could rescue by CO2, showing that they're actually not dead. They're just restricted in their ability to fix carbon based on a lack of carboxysome. But once we elevate the CO2, we can rescue their growth. So this is showing this kind of dynamic movie where the cells can grow very fast initially, and now they're growing slower because their carboxysomes are degrading and losing activity. At 42 hours, we had high CO2 back, and now, the population is really fully recovered. Okay, so with that kind of introduction to this carbon concentrating mechanism, which is really, a major passion of the lab, and I got involved in this, interesting DARPA project on engineering living materials, which was interested in calcium carbonates. And it turns out, my lab is very good at carbon fixation and understanding this. And it turns out a byproduct of the carbon concentrating mechanism is thought to lead to an alkalinization of the media, basically through a drawdown of bicarbonate faster than it can re-equilibrate with the media, and leading to this basically elevated hydroxide concentration outside the cell, which shifts the equilibrium of CO2 to carbonate, leading to carbonate precipitation, basically calcium carbonates. Other organisms are known to also do this through urea hydrolysis, so many soil microbes can do this. I won't have too much time to tell you today, but I will tell you that Synechococcus 7002, the organism we work on, has a carbon concentrating mechanism, but also has a functional urease. It hasn't really been appreciated that cyanobacteria actually have both of these pathways, and we can independently resolve the contributions of calcium carbonate production with urea and CO2 and combined in cyanobacteria. So I think this is something that's important, that you can genetically delete both of these pathways and they both are important carbonate producers in cyanobacteria, whereas the CO2 concentrating mechanism is thought to have been the dominant version. So if you look at kind of, these native environments, we often find, even today, cyanobacteria producing these carbonaceous structures like stromatolites. If you were to section through these, you find these, strata of, biomass and different carbonates, which are, these beautiful kind of layer by layer stone structures preserved in the geological record. These calcium carbonates are formed in many different types of crystal forms: trigonal, orthorhombic, hexagonal. We typically find these calcite, aragonite, vaterite, and these have a stability, a thermodynamic stability, such that there's a maturation and a dynamics of these crystals. And often, we find all of these different types of structures, but often it's manifested eventually in the form of calcite, the most stable polymorph. This is probably, you guys are probably experts in a lot of this stuff. I won't go into this, but I think, cement industry is this huge CO2 releaser. Could we actually harness the properties of nature to basically make these carbonates and not actually have to extract them from nature? So very simply, we found that just by growing cells in elevated carbon and calcium carbonate, with elevated calcium chloride and sodium bicarbonate in solution, basically providing a supersaturated condition, the cells will rapidly induce extracellular mineralization of calcium carbonates. Here you can see these little tiny dots are cells, and these are, biogenic minerals, and I'll show you some pictures that these are associated with cells. And it turns out, if we look at these by Raman imaging, we can actually see in a single condition, we do get the majority of the structures. We get our calcite, but we can also get aragonite and vaterite under different conditions. Notably, if we just change the salt conditions or the media conditions, for example, the crystals are extremely plastic in terms of their morphology. This is known just based on, other, basically abiotic mineral precipitations. By changing the magnesium and the calcium ratios, you can, change from kind of more square-like crystals to more, round-like crystals. So it turns out, there's a fair amount of tunability, just by changing the conditions, even at the same kind of growth rate of the cell. For example, these cells can grow in really low and high salt conditions, for example. And it turns out that the crystals that we produce are very different than kind of the abiotic crystals. There is a fair amount of abiotic crystals that can be produced under these conditions because there's really a pH influence. So you can easily produce calcites by changing the pH. But the morphology is much different than the crystals you see on the top, where you can actually see these extensive casts left behind by the cells, which you do not find, and often the morphology is very different. Kind of importantly, I think one of the key features is that this requires this change in pH. So when you grow the cells, they will have a rapid change in pH in the light as they basically get carbon starved. And for the people studying these minerals much more extensively than me, we found that actually we found this very distinct brucite nucleation that was only really found under these photosynthetic conditions of calcite production. So if we inhibit photosynthesis with DCMU to inhibit photosystem 2, we change we inhibit this rise in pH. This requires photosynthesis, or if we put the cells in the dark. In this case, we do get some minerals. I would say this isn't really quantitative in yield because we're trying to enrich for SEM. So we get a lot less minerals, and many of these are like semi-biotic, I would say. But the key is that we do not see any brucite in these different conditions, which is kind of distinctive. And furthermore, we found that if we knock out the carbon concentrating mechanism, which is really thought to be essential for this process, we can form, even under elevated CO2 conditions, very distinct types of biotic minerals with a strain lacking the carbon concentrating mechanism entirely. And so Here's compared wild type versus the mutant. The morphology is different. You often find much more interface with the cells in the wild type. But I think this is substantial and could reveal some additional mechanisms in biotic calcites. Let's see. I want to leave a few minutes for questions. I'm going to just go through kind of a quick gallery because I want to show some of these pictures. Much of these are really unpublished and will be coming out soon. But I think the diversity of these crystals are really kind of fascinating for me. These are just by kind of tuning the environment of wild type cells. These are what I call the calcite hotels. You often find these kind of more disc-like structures. These will manifest into these square-like crystals where you can now see the cells highly interfaced and eventually forming facets. These are calcite. Under different conditions, you can actually see these kind of mixed crystal landscapes with aragonite and calcite here. This is kind of one of my favorite crystals once you start seeing how interfaced. And one of the important things is we've worked a lot with other bacteria and even engineered this into E. coli. E. coli will always cast itself and die. Bacillus casts itself and dies. Cyanobacteria always leave an open face sandwich here, allowing themselves to thrive within these crystals. And actually, part of this DARPA project was to prove viability in these structures. And really, this is a happy place for cyanobacteria. This is a protective environment. I think this is very substantial for evolution. I'm just going to show some of these really high-res early nucleation structures, showing, for example, this aragonite fan transient polymorph, that we often see, formed very early on. We can see calcite starting to form on the surface of cells in these different conditions. Under these different nucleation conditions, we see a lot of these unusual structures with very highly interfaced cells. So I'm just kind of showing this more to, maybe spark some conversation. But really, the key is you can see very highly interfaced cells with even, these fibrous types of carbonates. But what you'll notice is that they're not trapped. These are actually seem like they might even be able to, actually demineralize these and actually, through acidification, for example, mineralize and demineralize, for example. I'm going to highlight this crystal because I think this is one of the most interesting and kind of, I think, substantial ones where if we look at this, this is a pure calcite matrix. Each one of these little divots is a cell. This grows by layer growth, almost like a coralloid-like structure. If we were to zoom in, we can see each one of those is a cell. These never get trapped. It's very unusual because you'd think that if you get rapid growth of these calcites, it's going to cast the cells. They never cast, suggesting that this is really a process that's controlled by the cell. So let me just say this data was from many years ago. And I've always been wondering, how is this working? I never really understood what the mechanism by what this is formed. Recently, after several years of work, we've now been able to figure out how to combine our live cell cyanobacterial imaging, which has kind of been the limiting factor because it takes tens of hours, requires light CO2, and the simultaneous imaging of the biogenic crystal growth in the same environment that we produce this crystal. So this is basically, we finally got the film of this being formed in live cell imaging. And it's actually, I would just tell you that it's totally made me rethink of how these are formed. And I think this leaves the field open in some of the questions. So here we can actually see there is a substantial amount of these small abiotic crystals, these little barbells of calcite that we can actually see that are often formed. These are similar sizes to the cells. And here I'm now going to start this movie. So what you can see here is a rapid growth of the crystals. And now I want you to focus on, if you can see my mouse up in this corner. Here now the crystal is casted a cell. So the crystal is now grown into the cell, casting the cell. It wasn't actually nucleated on the cell. And now actually this mother cell is trapped in the cell, and all of its daughters are just being buttered out. This cell never can escape. And this is actually leading now to this high local concentration of cells intricately trapped and associated with the crystals. You can actually see this. And this is kind of now just capturing, I would say, the phase 1 of the nucleation site. Once we form these structures, those structures then actually will lead to then this kind of more long-term growth, where first you form this rigid interaction between cell crystal. The cells are casted as they're trying to escape and grow out of this crystal. You actually get leading edge growth of crystal behind them, recasting them. And so that is like a maturation process. And so I think it's this changed our idea on, you know, where it's nucleated. So with the last minute, I think I'm probably over time. These cells also just grow in sand, it turns out. So we can just, instead of growing them on a petri dish, this is just growing them on media with sand over time with this calcified media here. This is them growing on just Ottawa sand, pure, like a quartz sand. These are cells that will just grow and form a biofilm on sand. They will actually form calcite and bridge these sand grains together. If we remove the cells, we can look at these calcites left behind by the cells. You can actually see they're all casted by these cells, actually. And if we were to, you know, look by EDS, for example, we can show that these are actually calcite on silica matrix, you know, basically biogenic. You know, these are individual traces of these crystals. And this is just kind of a picture of now what you can do with this, where you can just now take sand, put it in any arbitrary mold, add the cells, let them induce this precipitation, demold it, and just dry these structures. And this is actually a masonry grade concrete beam here that is still living. And so you can actually break this beam here. So add living materials once, take this beam, break it, and then actually have a regenerative cycle where we've done this over about 4 or 5 generations to just grow bricks from bricks with only a single inoculum, because we've maintained them viable. So this has been a kind of an interesting joint project with a civil engineer or 3 civil engineers. This was, you know, featured and got a lot of press, which is kind of interesting. But I think this is a potential way to have, you know, displace a lot of the carbon in the cement industry. With that, I'm gonna just thank all the people that did this work. I mean, I would say a lot of this work was done by a really good student, Nick Hill, who's just about to graduate now. You know, my geology and civil engineering colleagues, DARPA for funding, and I will maybe stop share and take any questions. And thank you for your patience.
Thanks, Jeff. We are open for questions. So anyone either can have a question, can put it in the chat, or you can just raise a hand and I can unmute them.
And sorry, I went a little over, but I have plenty of time if you have any questions. So.
Yes, I have one question. Thanks for this very nice talk. My question is about the tool that you present, Deep CYAN. Did you already try to use it on environmental samples, or is it only for axenic strains?
Or so on filamentous samples, you meant?
Yeah, or a mix between several communities, for example, from environmental sample and not culture ones.
Yeah, so we've done a lot of things of just mixing strains purposely together in different mutants and making defined mixed communities. It's very good at that because we can just film everything together, you know, under the exact same conditions, and then computationally segment them by trait, by cell size, or initial fluorescence. So you can, you know, easily use this to look at mixed microbial communities. We're doing this a lot. We're also looking at a lot of filamentous cyanobacteria, so these Anabina strains and ones that differentiate into complex morphologies, and, you know, environmental samples. So I think it's really the main thing that my lab is focusing on now, because once we figure out that it's working, it's too exciting not to do it because it's so fast. It takes the processing from, you know, 2 weeks to 1 minute, and really and really the key is just getting the good ground truth data set. So if you have like any mixed community, you know, you could just do kind of like a static segmentation and then train the neural network with that. But I'm happy to discuss that. Those tools will be online, but I think the ability to now just make rapid progress using that is and it will just get better over time. The good thing is if you're really good at storing your database and storing everything, as you use the neural network to segment, you'll get actually better and better, and you can like iteratively use that to like retrain itself over time, and then reanalyze old movies and then reuse those to feed back in. So actually, like if you set up like a pipeline that can reanalyze data over time and feed it back in, like it will just get better and better and better, and over time. That's the good part about it. So,
Karim, you have a question?
Yes, thank you, Jeff. That was really fantastic. I really enjoyed, and I guess I would have like 10 questions that I note down, but I keep.
We'll follow up by a Zoom too. So ask your important one, but we should just go touch base anyhow.
That would be nice. So I'll pick only two of them. So in the last over the last slides about these cells that get trapped by the precipitation of carbonates and then divide. I was wondering, so I could not see that on your movie, the cell that is trapped by the carbonate, is it still, so it's dividing once, only once, or is it dividing for a longer time?
It's dividing a lot. So I don't know if you've ever seen in E. coli, and people have made what's called a mother machine. It's a microfluidic device where you can trap the mother cell, and then every cell divides and washes out. This is creating a mother machine. So the mother cell is trapped, and it's basically putting out dozens and dozens of daughter cells. So it is very metabolically active, and it is basically producing all of those cells. And it's interesting because in other cyanobacteria, people have actually found that, you know, because there are cells that are interact with rock and might eat carbonaceous rocks, and they will often express these calcium ATPases on their surface when they're interface and actually differentiate in kind of a different cell type. So, you know, it's very possible, you know, once you're trapped, you actually go under kind of a different differentiation because you're you know, now interface with this different, you know, completely different you know, surface. And so that, you know, goes to be shown, you know, is the physiology of that mother and the daughter cell, you know, very different, but they're very active and they're very alive. So they're producing a lot of cells, yes.
And so do you think their photosynthesis activity, for example, might be even higher than the free cells, or?
That could be possible. I think, so now, I mean, we probably have that data where we could look at the growth rate difference in these lineages. I mean, we actually have that data. I haven't really totally looked. I'm really interested in that. What I've been particularly interested in is, you know, the growth rate, but also just the fitness of these cells, and what is the selective advantage to be mineralized, because in some cases it could be lethal, because if you're in the ocean, this could make you sink to the bottom. And as the farther you sink, the darker it gets and the harder it is actually to get back up. It's exponentially harder the farther you go, because you get more energy limited. And so if you go too deep, you can never come back up. And so these are often found in these like cementing sands in the Puerto Rico, like northern Puerto Rico, Caribbean. You have this like carbonaceous rich, deep Atlantic Ocean. It upwells. And so like in the Turks and Caicos, this is where you get these ooids and these carbonaceous sands. Puerto Rico, it doesn't go to the other side. So you have like cementing sands. So I think this is probably much, and like Seneca 7002 is found there. So this is likely more of like when you're growing on sand and these biofilms. But the one thing that's been very interesting is that, you know, I think it could be very important for protection from high light environments. You know, and in that case, in a high light, you want high CO2, because if you have a lot of light, you need, you know, a sink for that, which is carbon. And the carbonates actually might create a shade and even the optimal light for photosynthesis. You know, this is something that I've been thinking about. I'll share this to you guys, because I would say I have too many things to do. And the thing that's been I've been very interested in these manganese calcites. And manganese calcites, these like rhodochrosites and things, where manganese is a key element for photosynthesis, for oxygen evolution. So is calcium. It forms the water oxidizing complex. So is carbonate. But it turns out magnesium calcite has a very interesting optical property, that it actually fluoresces, it absorbs UV and fluoresces at 630 nanometer light, which is the exact wavelength that cyanobacterial like. And so it's a very odd thing where it's basically it's a down conversion of UV to red light in conditions where the cells are under evaporating high light conditions on the sand. And these cells are known to be very high light tolerant. And so it's basically like when you're evaporating, and the interesting thing is if we just evaporate our media, which is salt water, which is, you know, over 300 millimolar sodium chloride, if we evaporate them, they go into calcite and not halite, which we do produce a lot of salt crystals, but they're partitioned into calcite, which is kind of interesting. So you can, and these are found in these tidal mudflats. So as the tide goes out, you're in bright Caribbean sun, you're evaporating, it's raising the carbonate levels because you're just evaporating water. They nucleate this on themselves, which will, could provide this protection from this high sun when you're basically now on the sand. And, when you go back into the, to the ocean, it can redissolve and now you have access to CO2. To me, that seems like a very likely kind of like day-night cycle where it's this interaction with this calcite, it's protecting you. So now we have a really good system to actually measure UV and like photo damage and like this high throughput photo damage experiment. In the microscope, now we're applying this to like in the crystals, are the cells actually protected? But long story short, I think this is going to be important of like a UV protection mechanism that might have even been important for like the evolution of photosynthesis in general. So.
Thanks.
Just a related question to that while people are thinking about questions. Did you, for the there was 7002, how they are attached to the mineral substrates, did you check about whether they have some sort of substrate selectivity? So for example, if you have apatite, like a calcium phosphate, and if you put it in the culture, would you expect them to colonize, like basically attach to, like form that mother cluster which you're talking about in the similar fashion? Or is it very specific to carbonates?
Yeah, so with 7002, we haven't really done those experiments because it's so they're non-motile. And, it turns out it's kind of complicated if you just randomly put things together and then statistically look at interactions. It's hard to tell, like, are they moving or preferentially enriched? Because, just because we see them next to it, we don't really know. So we started working on these other natural strains that we extracted from ooids. So these carbonaceous sands in the Caribbean. It turns out that like we've been able to enrich cyanobacteria from them. They're motile and they swim and they're very fast. And so it turns out like it's a much better system to look at like what cells they like, because we can basically like put different minerals and film them and see like are they actually like, is there like are they moving towards them, away from them, do they inhabit them?
Right.
So I think those are the questions I'm really interested in, that type of like, are they going and seeking them? Is there kind of this chemotactic? I think just having the right system, because like these cells will just expand and grow and eventually will hit it. But like setting up a system where you can actually like allow them to, choose this one versus this one and actually tell is a little bit harder. But those are like the great questions, because I think you'd imagine, like even for example, like under low manganese conditions, where manganese is essential for photosynthesis. I'm really interested in if I just have manganese calcites, you know, and just like have like this basically where I have manganese starved media, no manganese, and then drop a manganese calcite with cyanobacteria. Will I just get growth like in some sort of little cluster around that? Could that, you know, do you get localized growth? Those are the experiments I'm very interested in, but have not done yet.
Yeah, no, that would be super cool because it also will give some insight into like whether this is some sort of like not specific to any substrate, and they just, the cyanobacteria just likes a substrate, which is whether it is a passive or active utilization, it's just gonna stick to it because it's present. So it would be something very interesting to know. I'm gonna hold my other questions because I have 3 other questions. Elodie would like to have another question. Elodie Dupret, so she has a question saying, also, did you observe some specific patterns of expression and/or activity of bicarbonate uptake system, example, big A protein, according to the localization of the cells within the community?
That's a really good question. We have not looked at, so in a microcolony, in a single layer 16 cell microcolony, it's not nutrient limited until it goes into another dimension. So it's on a cover slip and we're giving them bicarbonate, you know, at millimolar levels. And so there's a huge mass of bicarbonate in that 2D level, you know, so like they get mechanically confined in the middle before they get, you know, carbon limited, for example. And we spent a lot of time, you know, kind of, you know, proving that. Once cells go into 3D layers, then it's likely, I would say, almost certainly they're going to be carbon limited on the inside. This is going to change the expression of the transporters and everything like that. We have not really looked at that. I think that's a great idea. The you know, the transporters themselves are, you know, there's a lot of mystery, and you know, when the cells grow in high versus low CO2, their own affinity for CO2 changes. You know, we're doing, you know, some work on the NDH systems and things like that, but yeah, I haven't really looked, but I would imagine yes, with caveats under different conditions. I could see, maybe I'll look at the next question. Have you had a chance to look at changes in cell ultrastructure or protein expression for the mother cells trapped in the crystal? I would say not specifically. We have a lot of tools to do this, and we might actually have the data because we have a lot of our strains have markers that we can look at protein expression. We have looked at, and we have a paper in Nature Microbiology looking at cells in mechanically confined environments, and so we know a lot just about the general principle of if you confine a cell under stiff media or anything like that, you will change protein expression. So this is like a very similar condition, with the caveat that I have no idea what the, you know, the cell interface with the mineral looks like, and are they taking up, you know, can they even use this as some sort of, you know, energy source or something? So I think that's a very, very interesting idea. The thing I'm most excited about, so we've been doing a lot more cryo-electron tomography, cryo-fib milling. You know, I didn't talk about it, but now we've been able to do like, you know, cryo fib milling and cryotomography on the carboxysome assembly pathway, and basically trapping cells under these different conditions. So we're getting really good at that. We just got awarded to be a cryo-electron tomography service center at CU Boulder in my building, with the fib milling and everything. We also have, you know, a lot like the Themis for doing a lot of elemental analysis. Over the next 5 years, my goal is to really start, you know, now that we have the tools to section through cell crystal interfaces, really get the cryo-ET structure of that interface and elemental maps, and really see what that looks like. I've no idea. That's like my dream, is to take one of those beautiful calcites, mill it, you know, down to an 80 nanometer section, do a full tilt series of what that looks like, and then bring that sample, which is going to be damaged, over to the Themis, and then do a correlated elemental mapping of the crystal, and have that structure. So that's, you know, the goal over the next 5 years, to generate that image. So. Well,
that was my question, and thank you so much for that answer. That, I mean, the talk was fascinating, and you may be hearing from me by email because I'm working on a different system. It's halophiles, halophilic archaea in halite, but it's a similar process. So I may be in touch with you because that sounds fascinating.
Yeah, I think now that these, the good fib mill and the cryo fib milling systems where you can just, we've done it a lot, and it's basically, I spend all of my nights zooming around through these, basically thick sections of individual cyanobacterial cells. Yeah, and it's pretty much unbelievable because you feel like a scuba diver in a cell. That is actually, I feel like, how we'll figure out how things working, because you basically want to just correlate the live cell and then just go look at it with your own eyes. I don't know. I don't even know if there's people on here yet, but I'll just kind of show the sneak peek of the type of data you can get out of this. So this is like a single carboxysome, and each one of those little marbles now is every Rubisco segmented. These are the assembly intermediates, the polyphosphate, every ribosome, but this doesn't even really kind of explain the depth of this data, because each one of these sections is 0.3 nanometers, so you get 10 stacks through Rubisco. So you can really actually see every single protein in the cell here at near molecular resolution, and the goal is to segment out every cell, or every density in this cell, and start looking at where things are, where are the crystals. But I would say it's so exciting to be able to look in the cell and see an individual Rubisco, an individual protein that what it is. I think the most powerful thing that we found is that a lot of people are just looking at things, and if you don't have the correlated map, you can just see anything you want. And it turns out that, like, using, I would say I'm a synthetic biologist, and we do a lot of stuff on that. I'm almost just have stopped doing a lot of like cloning synthetic biology, because the power of the environment is much more powerful, and to just synchronize things. So, like, for example, like, starve cells for manganese, add manganese, you synchronize the system to grow, and then just take time points. Or, starve them for calcium, add calcium, and get crystals to grow. And so, like, we spend a lot of time in like the synthetic biology, which takes years, and every clone costs like $20,000 in reality. And it's with machine learning and imaging, it's almost like there's no point, because you just use nature and the diversity. Just like take your cells under like 10 different conditions, and then you can basically like figure out like the principles of that just by using environmental poisoning. And so like almost everything, it's like with the cryo, it's like, just figure out how to like stall your cells to grow, induce growth, and then you can just trap like, crystals forming. So yeah, love to follow up. This is an exciting time. So I could talk forever about it, because it's like, once you get the picture, it's insane. Because one of these things, I'm writing 5 papers from this one data set, because it's just, the goal was to look at carboxysomes. It turned out we've been able to find the thylakoid membrane biogenesis, because when you turn on carboxysomes, it turns on membranes. And so, like, the side project is we basically found the nucleation set of thylakoid membranes. We also found all these, so almost you find so many other things. You need one data set, and you can just spend your life on it. So it's cool.
We have another question from Melissa Brito.
Hi, Jeffrey, and thank you very much for your presentation. It's a very, very nice topic. I'm also working with the cyanobacteria. So I have a few questions about your general approach in the lab experiments. Just wanted to ask you if you work with the pure cultures. Was that Synechococcus, if I understood well, right?
Yeah, this is the Synechococcus 7002. That's one of the pure culture. We have an entire culture collection in the lab, so we're looking at lots of unusual cyanobacteria, mixing them together, taking things out of ponds. Basically just like seeing anything we can film, and then can we extract the movie. So it works very, yeah. So you can do any sort of mixing of things.
All right, and So you grew the cells, you cultivated the cells, and later you showed the crystals that you obtained, the calcium carbonate crystals. I would like to know if you extract the EPS, because I didn't understand well how you precipitated it. Was it cells and EPS, or just EPS, or on the cells? Because I saw that you used the ammonium source and calcium chloride, but you performed on organic metrics or just on the cells? How was that?
Yeah. Well, so far, almost all of the cyanobacteria that we've tested, Seneca cystis 7942, if we grow them under this particular condition of this, almost all of them will induce biogenic calcium carbonate precipitation. Most all of these cyanobacteria do make an EPS, and they have some of them have extensive EPS. And I think the interesting thing is, in some conditions you can actually see mineralization on the surface of the cells. In other conditions we see this more of a casting of the cells. And so it's condition dependent, because if we grow cells in high or low CO2 or changing on the salt, there's a lot of differences. So I would say it could be very condition dependent. The EPS is likely very important, and a lot of these strains that we look at have really extensive sheaths, and we can see a lot of interactions, and eventually will get sticky. But it's not an absolute requirement, and under conditions of low carbon conditions, often they might not be producing that much EPS. So I think it's something interesting, but the physical interaction I think is still a little bit different. A lot of the high res SEM, we could see a lot of minerals on the surface, and so I was actually very surprised by the live cell image, where it's different. But in some sense I believe the live cell imaging a lot more, because preparation of dry crystals is prone to a lot of dehydration artifacts. I don't necessarily think those are. We're very careful when we look at stuff like that, and we do critical point drying and things. But anytime you're out of native, it's going to be my preferred way of even doing anything is taking the liquid and then just doing Raman mapping to just see by Raman in solution what are all the crystals, because then you're looking at the native system, and you can tell but with Raman organic material versus cells. And if I don't see the same thing by the SEM that I saw in the Raman, then I'm very concerned about the prep, because the harvesting and this is super salty seawater.
Right.
So if you don't wash your crystals right, you will 100% get crystals, and you can get calcites. If you just dehydrate things, you'll guaranteed get them. So yeah, I think there's a lot of ways to do that, but using the Raman mapping and then is the most useful.
Okay, thank you.
We have a question on the chat. Jeff, can you see the chat? Is it visible for you, or?
Yep.
Okay. So it's a question from Cecil.
Let's see. Concerning deep cyan, did you use a way to keep your cell in 2D? As some cells can actively swim, it would be difficult to track them. So, the easiest way is to restrict cells in a 2D layer by putting them in an agar pad or a microfluidic device, so they're forced to go 2D. The movement 2D is not as an issue as 3D. If you're doing wide field imaging, if they go out of focus, that's an issue. If they're moving and your frame rate's fine, then you can track them fine, because you can create, you're basically just doing a time series, image, image, image. So each one's independent. If you can get a mask, the next mask, if the cell is swim really far away, then that means your sampling frequency is too slow. And what you want to do is make sure that, they don't go too far away. And then you can use this distance matrix to determine, was this cell likely this cell in the next image, to link them together. So it's basically a cost distance minimization algorithm based on distances of all the cells. It's in our CYAN paper. It's a very nice thing. So in 2Ds you can just track everything by this distance. In 3D, it is more, everything's more difficult in 3D and then in 4D. But I think, yeah, you can overcome it. Just try to figure out like how fast you can image your cells without killing them or damaging them. Because like sometimes if we over image our cyanobacteria, they're sensitive to light, and that can lead to damage. So you want to make sure that you're imaging them without killing them, and kind of figure out that time scale. If these are non-photosynthetic cells, you could probably image them at like every 40 milliseconds, and then that's fine. So, yep.
I have a quick question. So I was looking at your the first part of the talk, which was all about like how if you grow a batch culture, and there's such a huge heterogeneity in the microbial population, and that's why single cell analysis kind of clearly show that. But knowing all that now, I don't think everyone can do single cell analysis every time when you want to see the impact of let's say a single environmental factor, right? What do you think are the changes things we should keep in mind when we're setting up a new culture in order to have a more homogeneous population and minimize any sort of those kind of heterogeneities? I don't think we can eliminate them at all.
Yeah.
A batch system, but what are the few things we can keep in mind?
Yeah. One thing we've been doing a lot with that doesn't, it's still kind of single cell, but doesn't require any of the time series imaging, which is much more complex, is because 7002 is so reproducible with so much red light in 16 hours, it will always produce from a single cell that 16 cell colony. And so I can have everyone in my lab just study 7002, and I can look at everyone's data and I can tell if it's messed up, because if it's not producing that shape of colony, I can actually tell, like, if it's asymmetric, you can tell like, oh, a cell must have died in the 3rd generation to produce that colony. And if they show a video and they never reach it, you can tell it's because when people are making their slides, if the agar is too stiff, that represses it. And so if you have stiff agar, it represses that growth. So I can immediately tell what they should look like. And then you also know, like based on the position, all this info. So we can do a lot of information where I just have people take cells from a pre-culture and then just spot them on slides under, 50 different conditions. And then just don't even grow them in the microscope. Just put those slides in our chamber, grow them up for 12 hours, and then just go take an image of all of them. And it's now you can like directly compare like your pre-culture, what they should have on your control, that they should have grown to a 16 cell colony. The morphology of the colony, everything is important. And you can basically take all of that data, and that is sufficient to build like a machine learning database to now like say, here's a batch culture, the cells look weird. What nutrient are they starved for, right? Because now you can like train it on individual starvations, for example. So those types of experiments I think are very valuable, of like just like screening conditions and making these like factorial analysis. But then in batch culture, I think just like basically thinking about going eventually a lot of times you need a lot of biomass, and you're not going to you need to do batch cultures and things. It's been interesting, like to find phenotypes in single cell and then figure out how to get a lot of it in bulk. And we can go that way. We found like unusual, like the degradation center for the carboxymes we found in single cell, and then use that to identify the conditions where we make them, and then scaled up, found them in bulk, purified them. And that was like a single cell needle in the haystack to enriching from like 1 in a million cells to 1 in 100 cells to a bulk purification. So I think those, but also like the power of the turbidostat, I think is like really important, of growing cells under a continuous dilution, where there's still some challenges, but if you grow cells under a continuous dilution, then the light levels are the same. And I think focusing more towards like, steady state and like thinking back to, Minode, who's probably, has some little plaques on the wall not too far from you guys. And basically thinking of like the principles to understand growth requires this steady state condition, which is never really achieved in batch cultures of cyanobacteria. And, my friend Ryan Clark made a really good model of like light limited growth and like understanding that process under different configurations. And so I think really like just considering the light and how that will influence everything, and the carbon, and like knowing what your limiting resource is going to tell you, like, are you light limited or carbon limited? If you don't know in your experiment, that is the danger, because it could be changing, and you want to know, what is your limiting nutrient. That's going to tell you a lot about what's happening. So, all right, I might have to jump off, but I wish I could just stay in Paris forever and talk to you guys, and hopefully I can, I will definitely go there, and I'll stop by in person one of these days. But I would just say I'm totally open for collaborations, follow up. We have a lot of tools. I'm very slow to publish because I just working on too many things at once. I have 4 little kids, so I'm very happy to share unpublished data and other things. That'll be much faster, because I think it's a good community, and there's a lot of like important things that we have to do with cyanobacteria, and it's really could be synergistic, because I think there's too much important things to do and not enough time. So, all right.
Well, thank you so much, Jeff, for a fantastic talk, and really, really great talk. So
all right,
I think many of you will be in touch, I guess.
Yeah, I'll follow up with a lot of you. All right, bye.
Bye.
Bye. Thank you.